So I'm going to talk a little bit about photosynthesis, but let me just point out that I'm not going to talk about everything that you need to know about photosynthesis. There's some things that come straight out of the book, some very basic ideas about wavelength, spectrum of light, um, what can happen with light, and there's also a Prezi posted up on our Blackboard page that kind of goes through a little bit of that. So I direct you there. But what I want to talk about here instead are two important parts of photosynthesis. But again, I'm not going into all the detail that we know, just enough for you to make sense of what's going on without overloading with too many details. Let me just point out, let's break this word down. So photosynthesis, you should know photo means light, and synthesis, now you know, means is a process of making something. So think back to our synthesis reactions, our dehydration reactions, condensation reactions, all the same thing of making something. So we're using light to make something in photosynthesis. So that being said, let me just give you the overall equation for photosynthesis. It's water plus carbon dioxide. I'm going to put a light over the arrow here because it's a form of energy. So we got water and carbon dioxide plus light, giving us C6H12O6 carbon dioxide plus oxygen gas. And this is the net reaction. This is what goes in and this is what comes out. But there are a couple of intermediary products and reactants, and I'm just going to list them here and we'll come back and talk about them. There's NADP+, which converts back and forth from NADPH, and we've got ATP that goes back and forth from ADP plus a lone phosphate group. And so these keep going back and forth within our processes of photosynthesis. So these are molecules that we'll talk about in just a minute. And I'm going to just do a quick diagram here as best as I can. So this is supposed to represent my chloroplast. Throughout the chloroplast, we have thylakoids, which I equate with like pancakes. So each one of these is a thylakoid. The stack of thylakoids is called a granum. I'll just write these words up here. I don't want to clutter my space here yet. So each one of those pancakes is a thylakoid. The stack of them is a granum. So the plural would be grana. And then all the syrupy stuff here is called the stroma, S-T-R-O-M-A. And know that the chloroplast has lots and lots of these thylakoids, and think about why they have so many of these small pancake-shaped things. It's because it gives it more surface area, and really this membrane, this thylakoid membrane, the outside of each pancake, the edge of every pancake, is really where the reaction occurs that we're getting ready to talk about. So reaction 1 happens here in the thylakoid membranes, and reaction 2 happens here in the stroma. So go ahead and jot this diagram down, label the diagram, and then I would copy over another fresh diagram without labels, because we're going to add some notes on top of this chloroplast in just a second. And I guess one thing I want to point out before I go too far is back at this equation of water and carbon dioxide, glucose and oxygen. Just want to point out that glucose and oxygen are what we need for aerobic cellular respiration. So really photosynthesis is the process that makes our food, and plants also go through cellular respiration, but they're making their own food. And so by definition, plants are autotrophs, where we are heterotrophs because we have to consume these two things. And I want you to think back to which systems, which body systems in humans get access to this stuff. Glucose comes through our digestive system, oxygen comes through our respiratory system, and they combine in the mitochondria, or in the cells. Glucose gets broken down in glycolysis into our pyruvates, and oxygen gets used as an electron receptor and hydrogen acceptor in the electron transport chain, giving us water as one of the products of cellular respiration. So we're working backwards here. So where do we get the sugars and the oxygen from? It's this process, photosynthesis. So let's go back to our chloroplast here. I'll pick a color that stands out. So one of our reactants is light. So I'm just going to write the word light. So light provides energy to the thylakoid membrane, and it actually excites the water that also is coming into the thylakoid membrane. So water is one of the reactants that's used here. So we've got light and water. And what ends up happening is that this light, by exciting the electrons in water, actually splits water apart, and obviously not in a balanced equation, in a non-balanced equation. Water gets split into oxygen gas and is given off. So in a way, oxygen gas is a waste product. So chloroplast doesn't want it, but remember, it is very, very important for aerobic cellular respiration for the high efficiency of ATP that are used later. But oxygen leaves the system here. We've still got our hydrogens that are part of the system. So I'm going to use this orange to refer back to these intermediary molecules. And what ends up coming off of this molecule, off of this reaction, is NADPH. And a good way to remember that NADPH is associated with photosynthesis is this P for photosynthesis. So NADPH comes off of this reaction, and I'm going to go ahead and label it as the light-dependent reaction. Now, if you know anything about photosystem 1 and photosystem 2, that's what's happening here in the thylakoid membrane. That's a detail I'm not going to go into here because I'm trying to give you an overview so I don't lose you in too much detail. But the NADPH comes off of this light-dependent reaction. It's actually carrying this hydrogen and electrons from the water. And it's also taking another molecule, ATP. So think back to adenosine triphosphate. You should know what that is. And then what happens here in the stroma? There's a series of reactions. It's a cycle. I don't want to give you too much of a drawing because we're gonna learn it as a. I'm just gonna put it as a box, and we're gonna call this the light independent reaction, also known as the Calvin cycle. And so those are both names that you need to know, both of those names. And one thing that you should also know is that if we look back at our reaction, we've got our water taken care of, we've got our light taken care of as a reactant. This is where carbon dioxide comes in. Carbon dioxide comes into the system here. Now remember, this is atmospheric carbon dioxide, and here's oxygen gas. So these things are coming in and out of the cells through the stomata, also known as stomata in the bottom of the cell of the leaves, allowing gas exchange. This water is coming up the root through the roots of the plant, through a traditional classic plant, and it goes up the plant through. I guess I'll tell you how to spell this. Through the xylem, which is vascular tissue, and it travels through capillary action. Remember, water being polar and creating hydrogen bonds, and that cohesive and adhesive properties, allowing the water to climb up a tree, in effect fighting the force of gravity. So water travels up, it's here, but it also can leave through the stomata through which the oxygen and carbon dioxide are entering and exiting the leaf. That'll come back into play in just a minute. But this carbon dioxide goes through what we call, I guess another term that we use, I'll use a better color, what we call carbon fixation. So the carbon in carbon dioxide, an inorganic molecule, gets converted to an organic molecule, glucose, C6H12O6. So the carbons came from the carbon dioxide, the oxygens and the glucose came from the carbon dioxide, the hydrogens came from the NADPH, and that hydrogen came from this water. So that's the essential reaction. But remember that we've got this molecule that's carrying the hydrogen, and this ATP is carrying some energy for the carbon fixation. And so what we end up seeing is another product coming off. The energy got used up from ATP, so that bond got broken. So we have adenosine diphosphate and the lone phosphate, and NADP plus. So it has given away an electron, so now has a positive charge, and has also given away its hydrogen to this carbon fixation reaction. So these two molecules are going back to this light-dependent reaction to get new hydrogens, new electrons, and keep cycling back and forth. So while we say this is the light-independent reaction, it is dependent upon the reactants here that came from the light-dependent reaction. So this part the light-independent reaction can occur when there is no light, but only for so long. And so we often see photosynthetic rates come down without light, because this light is providing the original source of energy that is then converted to the chemical energy of glucose. So this is what you need to know. This is the light-dependent reaction happens in the thylakoid membranes, and the light-independent reaction happens in the stroma. So I guess one thing that I want to point out is that any of these reactants, water, light, carbon dioxide, if any of those things are limited, we always see a curve. Draw as straight a line as I can. Whenever there's a limited reactant and we're increasing, let's say we increase, so if increasing one reactant, which implies that the others are at a constant rate or constant level, then we'll see a curve. If this is rate of, let's say we're measuring photosynthesis by oxygen gas, the amount of oxygen that comes off. But remember, that's a byproduct, it's a waste product. But let's say that's how we're measuring it. We would see a curve that looks like this. It levels off because no matter how much of this extra one reactant we add, we only have a limited amount of the other, and that's the limiting reactant. So that's what we see. So when we say we're increasing the amount of carbon dioxide or the amount of water, we're going to see a curve like this because we're not increasing the other reactants. But what's interesting, coming back to this idea of the stomata, that if we're increasing temperature, then if this is the rate of oxygen gas, what we actually see is a curve that looks something like that. And what's interesting about this is that as the temperature increases, the molecules are bumping into each other, they're going in and out of the stomata faster, and then at some point the plant closes those stomata. It's no longer allowing carbon dioxide and oxygen in and out, and by doing so, it's also preventing water loss, which is what it's trying to do. Plants are very dependent on water. They need water to do photosynthesis. They can't go get it. They can only grab what they can out of the soil. So they need to conserve it. And so what they do is close those stomata, preventing transpiration or water loss through their leaves. And so they shut that down. But doing so also shuts down the photosynthetic rate. So you should be able to explain these graphs. You should be able to draw them, identify them, annotate them, explain them, um, based on your understanding of this process. Now, one thing that I will tell you is that there's a reason I didn't put any values on the x-axis, and that's because I want to talk a little bit, and not now, in class, I'll talk a little bit about plant adaptations. So obviously not all plants are alike. We have different kinds of species in different ecosystems and different biomes, and plants have taken on adaptations in different environments. So our pine trees don't look the same as the cacti in southwest the United States. So there are plants that have taken on adaptations, have adopted or inherited adaptations that help them in these different environments. But they're still limited in this way by these reactants, but they're able to take advantage, I guess, of different environments in different ways. So we'll talk a little bit about this in ways that they can avoid limiting reactants or temperature or things like that. So, and I really, I guess, sorry, as I'm thinking about that, one other connection I should make. So anytime you hear anything about increases in temperature, think enzymes. So both photosynthesis and cellular respiration are controlled by enzymes. Remember, that's part of the definition, the controlled breakdown of the definition of cellular respiration, the controlled breakdown of glucose into ATP or usable energy. Well, those enzymes are what control that. So as the temperature increases, remember that enzymes become denatured. And those amino acids change structures, they misfold, and think about active sites and substrates. If those active sites change shape, then they won't be able to bond into the substrate, and they won't be able to move or drive the reaction forward, thereby shutting down the reaction, which is what we see happening here. And this is kind of going to be a very similar curve in anything that's temperature dependent in a biological system. Okay, so I think that I hope that makes sense, and know that there's a lot more detail about photosynthesis out there, and we're just very much scratching the surface here. Okay.